Welcome, everyone, to our high-yield review of Haemophilus influenza. Now, this is a really interesting bacterium. It's got a name that's totally misleading, and it kind of has a split personality. Sometimes it just causes a simple earache, but other times it can lead to life-threatening meningitis. So for your exams, you absolutely have to know the difference.
So here's our game plan for today. We're going to start by getting to know our suspect. Then, we'll check out its arsenal of weapons, we'll look at its different aliases, see what kind of trouble it causes, and then, finally, we'll corner it in the lab and figure out exactly how to take it down.
All right, let's kick things off with Section 1, Meet the Suspect. Let's get the basic ID of Haemophilus influenza locked in. Okay, so the absolute most important thing to know about this bacterium is that it's fastidious. That's a fancy word for being a really picky eater. And its name, Haemophilus, literally means blood-loving, which is your biggest clue. See, it can't just grow anywhere. It needs two very specific ingredients from blood to survive, factor X and factor V. Here's a little bit of trivia that loves to pop up on exams. Why is it called influenzae? Well, it's basically a historical accident. Back in the 1892 flu pandemic, a doctor named Pfeiffer found this bacterium in his patients and thought, aha, I found what causes the flu. Of course, we know now that influenza is caused by a virus, but that misleading name just kind of stuck. All right, so we've identified our suspect. Now let's take a look at its arsenal. What exactly makes this little bacterium so dangerous? Well, it all comes down to its virulence factors. Now, of all the weapons in its arsenal, one of them is the big one, the polysaccharide capsule. If you only remember one thing from this section, make it this. This capsule is basically the bacterium's invisibility cloak. It physically stops our immune cells from grabbing onto and eating it, a process we call phagocytosis. This single factor is the key to its deadliness, and as you're about to see, it's also how we classify it, and thankfully, how we vaccinate against it. So, the presence or absence of that capsule is the dividing line. It lets us classify H. influenza into its main forms, and they have totally different effects on the body. I need you to memorize this acronym, PRP. It stands for Polyribosylribitol phosphate. This is the specific chemical that makes up the capsule of the most dangerous strain, which we call H. influenza type B, or just Hib for short. Now, this PRP capsule is a real double-edged sword. On one hand, it makes Hib incredibly nasty, but on the other, it's the exact target we use to make the life-saving Hib vaccine. This table right here is pure gold for your exams. Seriously, you might want to screenshot this. It lays out all the crucial differences. On one side, you've got type B or Hib. It's got that PRP capsule, it invades the bloodstream causing really serious diseases, mostly in kids, and thankfully, we've got a vaccine for it. On the other side, the non-typeable strains, they have no capsule, they cause more localized infections, mostly in adults, and there is no vaccine. And hey, if you're in a time crunch, just burn these three core distinctions into your brain: capsule, invasiveness, and vaccine availability. That's the fastest way to keep these two straight. Okay. So we've got our two different culprits, the invasive encapsulated Hib and the more localized non-encapsulated strains. So let's go visit the crime scene and see the specific diseases that each one causes. Because it has that protective capsule, Hib can just cruise through the bloodstream and cause invasive disease. This means it can travel all over the body, causing really severe infections, especially in kids who aren't vaccinated. The big ones to remember are meningitis, septic arthritis, and the absolute classic for exams, epiglottitis, which gives you that dangerously swollen, cherry red epiglottitis. Now, the non-typeable strains are a whole different story. Since they don't have that protective capsule, they can't survive a trip through the bloodstream. Instead, they cause disease through local invasion, basically just spreading to the areas right next door. And this makes them the #1 bacterial cause of two super common conditions: otitis media, or middle ear infections in kids, and acute flare-ups of COPD in adults. Okay, so we know the clinical picture. Now it's time for the forensic investigation. How do we prove in the lab that H. influenzae is our culprit? This stuff is absolutely critical for your practical exams. You know, the investigation follows a really clear path. You get your sample, say some cerebrospinal fluid if you suspect meningitis. Then you take a look under the microscope, you try to grow it on some special plates, and then you run some specific tests to confirm its identity. So the first step is the gram stain. You're looking for gram-negative, which means they'll be pink, coccobacilli. But the real keyword here is pleomorphic. It just means the bacteria don't all look the same. As you can see here, you get this mix of little rods and some more round coccoid shapes. Okay, listen up, because this is a super important point, both for your practicals and for real life. This organism is cold sensitive. You should never put a sample in the fridge. The cold can kill it, and you'll end up with a false negative culture. You have to process it right away.
Now, on to culture. This bug is the perfect example of why we need different kinds of media in the lab. On a standard blood agar plate, H. influenza barely grows. Why? Well, factor X is available, but factor V is trapped inside the red blood cells. But if you use chocolate agar, which is really just gently heated blood, the red cells burst open, releasing both factor X and factor V. And that allows for the really nice, thick growth you see here, which is what you need for a positive culture.
And here's another lab finding that is a total classic. You have to know this for exams. It's called satellitism. See, if you streak H. influenzae on a blood agar plate along with a streak of Staphylococcus aureus, you get this really cool phenomenon. The tiny H. influenzae colonies only grow as these little satellites right up against the Staph aureus streak. That's because the staph is acting like a little helper, breaking open the red blood cells and spilling out that factor V that H. influenzae is desperate for.
And this is it, the final, definitive, confirmatory test. We've put little paper discs with factor X, factor V, and a combination of both onto an agar plate. And look, the bacteria only grow around the XV disc. That's our proof. It tells us, without a doubt, that this bug needs both factors to live, confirming that our suspect is H. influenzae.
So we know H. influenzae needs both X and V. But what about its cousins that only need factor V? Well, for your exam, just use this simple mnemonic, para-HIV. That'll help you remember H. para-influenzae and H. para-hemolyticus. They only need factor V. All right, so we've identified our suspect, we understand its weapons, and we've confirmed its identity in the lab. So now let's talk about the final step, neutralizing the threat through treatment and prevention. The treatment strategy really depends on the strain and how sick the patient is. For life-threatening invasive Hib diseases like meningitis, you don't mess around. You go straight for a powerful third-generation cephalosporin, like ceftriaxone. But for the less severe, local infections from non-typeable strains, the key thing to remember is that they often make beta-lactamase, which makes them resistant to basic penicillins. And finally, for prevention, if someone's been exposed to a case of Hib, we give them rifampin to stop the spread. And this all leaves us with a really critical question for the future. You know, the Hib vaccine is one of the greatest success stories in modern medicine. But by practically eliminating type E, we've kind of created a vacuum. Non-typeable strains are now filling that void and are causing more disease than ever. So the real challenge for the next generation of doctors and scientists is this. How do we tackle this emerging threat, this bug for which we currently have no vaccine?